Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1B for November 2023. Let's begin with the first question. Question 1. This food web comes from a woodland ecosystem. Which organism is the producer? The oak tree is going to be the producer because all food chains in this food web come from the oak tree. So the answer should be C. And next they ask which organism is a secondary consumer. Secondary consumers are like the ladybird, and then we have the blackbird in this food chain. We have the hedgehog in this food chain as well as in that one, and so on. So the ladybird is one I chose because the others were not secondary consumers. Next they ask which organism is both a secondary consumer and a tertiary consumer. If we go back here, we can see the blackbird is a tertiary consumer in this food chain, but a secondary consumer in this food chain. So the answer should be A, which is a blackbird. And in part B, they say a farmer is using a pesticide in fields next to the woodland. The pesticide is reducing the number of greenfly and caterpillar in the woodland. Explain what effect this may have on the blackbirds in the woodland. If the caterpillars and the greenflies are reduced, these are food for the blackbirds. It means the blackbirds are going to also reduce. More worms are going to be eaten because that is going to be the only food source that the blackbirds are left with. So here I say decreasing numbers of greenfly will lead to fewer ladybirds. Decreasing the number of caterpillars and decreasing ladybirds will decrease the number of blackbirds. Caterpillars and ladybirds are a source of food for the blackbirds. So the blackbirds will have less food to eat. So more earthworms will be consumed, and the blackbirds could decrease in number due to bioaccumulation of pesticides. And again, remember, they're using the pesticide, so bioaccumulation could occur, and many blackbirds could die. Next, they say describe a different method the farmer could use to reduce the number of greenfly in his crop. They could use biological control, meaning introduce more ladybirds. If you're trying to control these, increase the organism that feeds on the greenfly. If there are more ladybirds, then it means the greenfly can be kept in control. So introduce more ladybirds, and these are predators of the greenfly. They will eat the greenfly to decrease their numbers. So this brings us to the end of question 1. Let's continue to question 2. In question 2, a student investigates the effect of different concentrations of sucrose on potato tissue. The student first makes a series of sucrose solutions using this dilution table. So we can see the different test tubes from 1 to 6, and then the volumes put. We can see here when we use 20, we use 0 of that, and the sucrose concentration is 1. So basically the sucrose concentration is decreasing, but we can see the overall volume is going to be 20 because we see 20, 0. When we have 16, we should add 4 because when there is 12, we add 8. 8, 12, then 4, 16, and so on. So this should be 4. And they say complete the table by writing the volume of distilled water for test tube 2. Of course, it's going to be 4 centimeters cubed since the total volume is going to be kept to 20 centimeters cubed. In part B, they say this is the student's method. They cut 6 equal-sized cylinders of potato, each 5 centimeters in length. They dry the cut surfaces using filter paper, and then they measure the mass of each cylinder. They place 1 cylinder of potato in each of the 6 test tubes containing 20 centimeters cubed of 1 molar sucrose solution, 20 centimeters cubed of 0.8 molar sucrose solution, 20 centimeters cubed of 0.6 molar sucrose solution, then 20 centimeters cubed of 0.4, 20 centimeters cubed of 0.2, and then just distilled water, 20 centimeters cubed. Then they put a bung in each test tube and leave the test tube for 1 hour. They remove the cylinders from each test tube, dry the cylinders with filter paper, and measure the mass of each cylinder again. The first question says explain the reason for step 7 in the student's method. In step 7, they dry the cylinders with filter paper. This is to ensure that any water or moisture left on the potato cylinders is completely removed so that it does not affect the results. So I say to remove the moisture, presence of liquid would affect the resultant mass, so it would be difficult to compare the results. In part C, the table shows the students' results. Again, we see for the different sucrose concentrations, we can see the original mass, the final mass, and the change in mass. Then we see the percentage change. The first question says calculate the percentage change in mass for 0.4 molar per liter sucrose solution. So for this, we have to get the change. The mass increase from 2.2 to 2.3, so there is a 0.1 increase. So the percentage increase is going to be 0.1 divided by the original, which is 2.2 times 100, and we got 4.5%. So the change is 0.1 here, and this is 4.5. Comment on the effect of the different concentrations of sucrose on the potato tissue. So this is a question mainly about osmosis. If the solution is hypertonic compared to the cells, it means the cells are going to lose water towards the sucrose solution, and the mass of the potato cylinders is going to decrease. If the solution is hypotonic, it means water is going to move by osmosis from the sucrose solution into the cells of the potatoes, and therefore the masses of the potatoes are going to increase. However, if the solution is isotonic, then it means there will be no net movement of water, and therefore there will be no change in mass. So I say the potatoes gain mass between 0.0 to 0.4 mol per liter concentration. Again, this is because the outer solution is going to be diluted, or it's going to be hypotonic in comparison to the cell, so more water goes in. And then as the concentration increases above 0.6, there is a decrease in the mass of the potatoes. In this case, there is a hypertonic solution of sucrose, so water leaves the potatoes by osmosis into the sucrose solution. So change in mass is due to the movement of water by osmosis into or outside the potatoes. Movement occurs from higher water potential to low water potential. So this brings us to the end of question 2. Let's continue to question 3. In question 3, a student investigates the differences between inhaled and exhaled air using this apparatus. So we can see the setup. There is test tube A and test tube B, and there is solution X. Air is going to come in from here because this is connected directly into the solution. So when the air enters here, if you have maybe this is going to be a hydrogen carbonate indicator or lime water, we'll see a color change as carbon dioxide from the air is going to be able to react maybe with a hydrogen carbonate indicator or with lime water. If it's lime water, we're going to see tiny bits of being cloudy. And then the air without carbon dioxide is going to go through here to the mouth of the person. And then as they breathe out, the air leaves their mouth, goes into this solution. Again, this is going to have a higher concentration of carbon dioxide because this is exhaled air. And then CO2 is going to be removed, and the remaining air is going to go out like that. So there is going to be a higher concentration of CO2 here, and the color change is going to be intensified. So they say they use solution X to compare the concentration of carbon dioxide in inhaled and exhaled air. Give the name of solution X. Solution X could be hydrogen carbonate indicator or lime water. Explain the changes that will take place in solution X in test tube A and in test tube B. I said if hydrogen carbonate indicator is used, the test tube will stay orange because at atmospheric carbon dioxide concentration, the hydrogen carbonate indicator is going to be orange. Then test tube B will become yellow because there is a higher concentration of carbon dioxide in exhaled air, so the CO2 concentration is going to increase, causing the color to change from orange to yellow. If you had used lime water, of course, test tube A could remain clear or could have some little cloudiness, and test tube B is going to be cloudy. Next they say, other than concentration of carbon dioxide, state one other difference between exhaled air and inhaled air. They have different concentrations of oxygen. For example, exhaled air will have less oxygen than inhaled air. And in part B, another student investigated the effect of the duration of exercise on their breathing rate. These are their results. So these are the results in the table. They say give the independent variable in this investigation. The independent variable is the thing that we change. In this case, the duration of exercise is changing, so that is going to be the independent variable. And then they say plot a line graph to show the effect of duration of exercise on breathing rate. Join the points with a straight line. So with my graph, I drew the breathing rate on the vertical axis, the units of breaths per minute, and the duration of exercise on the horizontal axis, the units of seconds. You have to show the vertical axis and the horizontal axis clearly with a clear scale, and then the points were plotted based on the table and lines were used to connect. So marks are awarded based on the vertical scale, horizontal scale, vertical axis being labeled and having units, horizontal axis with labels and units. Then the points have to be correct, they have to be connected, and again you have to use more than half of the provided space. Next they say describe the effect of duration of exercise on breathing rate. As the duration of exercise increases, the breathing rate also increases until 180 seconds. We can see it here until 180 seconds from this graph. Between 180 seconds to 210 seconds, the breathing rate decreases by 2 breaths per minute. And then after 210 seconds, the breathing rate becomes constant. And again, remember, this question is asking us to describe. You just have to write what you see from the graph. And then they say give one way the student could improve the reliability of their investigation. They could do this by repeating the experiment at similar conditions and then calculating the mean. So this brings us to the end of question 3. Let's continue to question 4. In question 4, the diagram shows 2 samples of blood seen using a high-power microscope. One sample is of normal blood and the other sample is from a patient with a blood condition. So we see the sample with normal blood and the sample from a person with a blood condition. The person with normal blood has more numbers of erythrocytes. These are red blood cells, so it means their body transports more oxygen in comparison to the person with a blood condition. However, the person with a blood condition has more neutrophils. Actually, neutrophils, lymphocytes, and monocytes, these are all examples of white blood cells. Your textbook talks about lymphocytes, but it doesn't talk about neutrophils. However, you can see the shape of the nucleus of this cell, and this looks like a phagocyte, so this carries out phagocytosis. It's a white blood cell. So here we have more phagocytes, so a lot of phagocytosis is going to be carried out in the person with a blood condition. They have more monocytes. These also carry out phagocytosis. They have more lymphocytes. Lymphocytes produce the antibodies. They have the same number of platelets. Platelets are used for blood clotting. So they say state 2 differences between the normal blood sample and the sample from the patient with a blood condition. Like I've already said, normal blood contains fewer monocytes and neutrophils. Normal blood contains more erythrocytes. Then in part B, they say the line PQ shown on the diagram has an actual length of 25 micrometer, where 1 millimeter is equal to 1,000 micrometer. They say calculate the magnification of the diagram. So they want you to measure this distance from that to that using a ruler. When I measured that, I got 14 millimeters. But since 1 millimeter is 1,000 micrometer, 14 millimeters is going to be 14,000 micrometer. Magnification is equal to image size divided by actual size. So the answer is 560. In part C, in a healthy person, 1 centimeter cubed of blood contains 5.0 times 10 power 9 red blood cells. An adult has 5 liters of blood in their body. Calculate the number of red blood cells in the body of a healthy adult. Give your answer in standard form. From the information given, 1 centimeter cubed contains 5.0 times 10 power 9 red blood cells. And since 5 decimeters cubed is 5,000 centimeters cubed, in 5,000 centimeters cubed, we'll have 5 times 10 power 9 times 5,000, giving us 2.5 times 10 power 13 in standard form. So that was my answer. And in part D, using the information from the diagram and your own knowledge, comment on the likely effect of the blood condition on the patient. We saw the patient had more monocytes, more neutrophils, less erythrocytes, and more lymphocytes. So I say there are fewer erythrocytes, which are the red blood cells in the patient's blood. So their blood will transport less oxygen, and their cells will carry out less aerobic respiration. Their cells will have less ATP, and they will have less energy for activity like running. They will easily run out of breath. Also, their blood contains more lymphocytes, which are the white blood cells. They will produce more antibodies as well as memory cells. The other type of white blood cells, they contain more neutrophils, so they will carry out more phagocytosis. So this brings us to the end of question 4. Let's continue to question 5. In question 5, guinea pigs have either long hair or short hair. The hair length is controlled by the gene with 2 alleles. The photograph shows a guinea pig with long hair and a guinea pig with short hair. So this has long hair and that has short hair. They say state what is meant by the term allele. Allele is a version of a gene. Next they say a breeder did the following crosses. In cross 1, a male long-haired guinea pig with a female short-haired guinea pig. This produces 5 offsprings, all with short hair. If all offsprings have short hair, having short hair is the dominant allele, and since we're short-haired, we give them capital H if I take H to be the trait for hair. And these are going to have small h. A male short-haired guinea pig with a female long-haired guinea pig. This produces 4 offsprings, all with short hair. Because all offsprings have short hair, it means this short-haired guinea pig was not heterozygous, but it was homozygous in this condition. And again, this HH is for that part. In cross 3, a male short-haired offspring from cross 1 with a female short-haired offspring from cross 2. This produces some offsprings with long hair and some offsprings with short hair. Draw a genetic diagram to show the parents, gametes, and all possible genotypes and phenotypes of the offsprings from cross 3. Because they crossed offsprings from cross 1 and cross 2, they were all heterozygous, so the parental genotype is capital H and capital H. The possible gametes are going to be H and H, as well as H and H. And if this is my genetic cross, the genotype of the offsprings is going to be that, and that. And the phenotype is going to be 3 short-haired offsprings and 1 long-haired offspring. Next they say, calculate the probability that the first offspring born from cross 3 is a male with short hair. This is going to equal to probability of male times probability of having short hair. Probability of a male is 1/2. And probability of having short hair is 3 out of 4 based on the genetic cross. So 1/2 times 3/4 gives us 3/8. Next they say, some short-haired guinea pigs are homozygous and some short-haired guinea pigs are heterozygous. Explain how a breeder could use a cross to determine the genotype of a short-haired guinea pig. Since being short-haired is dominant, we would cross a short-haired guinea pig with a homozygous recessive guinea pig, which is going to be long-haired. So I said short-haired could be capital H, or capital H. They could be crossed with a homozygous recessive, which is long-haired. If the genotype is capital H, some of the offsprings will be long-haired, meaning they'll be homozygous recessive. And if the genotype is capital H, all the offsprings from the cross will be short-haired. In part C, guinea pigs stay completely still and do not move when they sense danger. Describe how this behavior has evolved by natural selection. Usually when these kinds of questions are asked, you have to talk about variation. Those that have the genetic advantage survive, while the others do not, and then reproduction occurs so that the genetic advantage is passed on to the offsprings. So I said a mutation led to variation in behavior of the guinea pigs. Some were able to stay still if faced with danger, while others couldn't. Those that were able to stay still survived, meaning they were not eaten. They reproduced and passed on their alleles to their offsprings. So this brings us to the end of question 5. Let's continue to question 6. In question 6, the diagram shows a human reflex act with some structures labeled. It is the withdrawal reflex, which allows a person to remove their hand from a hard object quickly. In part A, which structure is the sensory receptor? The sensory receptor is part L. This is where the stimulus is going to be detected, so the answer is A. And then in part B, they say which structure is the cell body of the relay neuron? The relay neuron is here and the cell body is going to be structure P, so the answer is going to be D. The next they say which structure is the motor neuron? The motor neuron is going to be structure R, connecting to the muscle, so the answer is C. And which structure is the effector? The effector is the muscle, is going to be structure S, so the answer here is a D. Part B says a nerve impulse travels 50 meters per second. Calculate the time taken for an impulse to travel 180 centimeters. The distance required is 180 centimeters, which is 1.8 meters. If it travels 50 meters per second, that is going to be the speed. So time taken is going to be the distance, which is 1.8, divided by the speed, giving me 0.036 seconds, and that is the answer. This is S for seconds. The length of the nerve pathway in the withdrawal reflex arc is 180 centimeters. The time a person takes to remove their hand away from a hard object is called response time. Explain why the actual response time is different from the time taken for a nerve impulse to travel 180 centimeters. Our calculated time from the previous part is going to be shorter than the actual time it takes for the nerve impulse to travel. This is because in our calculation, we've not accounted for delays that occur during nerve impulse transmission. Delays do occur in the receptor, meaning delaying to convert the stimulus into an electric signal, and then delays do occur at synapses and so on. So I say there are differences in time taken to respond to stimuli. Delay occurs in generation of an impulse at the receptor, and then impulses do not cross the synapse directly. Time is required to release the neurotransmitter and for the neurotransmitter to diffuse through the synapse and to bind onto the next neuron for an impulse to be initiated in that neuron. So this brings us to the end of question 6. Let's continue to question 7. In question 7, biologists classify organisms into different groups. 2 of these groups are bacteria and fungi. They say complete the passage about bacteria by writing a suitable word or words in each blank space. Bacteria are small single-celled organisms. They have a cell membrane and a cell wall made of peptidoglycan. The peptidoglycan is what makes the cell wall of bacteria. Some bacteria are able to use energy from the sun to carry out photosynthesis, but others are saprophytes and feed on dead and decaying organisms. Bacterial cells do not have a nucleus, but most of their genetic material is contained in a circular nucleoid of DNA. They also have small circles of DNA called plasmids in their cytoplasm. Some bacteria are used by humans in food production. An example is using lactobacillus to make yogurt. Other bacteria such as pneumococcus cause disease and so are called pathogens. In part B, a microscopic single-celled fungus, yeast is used in the production of bread. Describe how yeast is used to produce bread. To make bread, we need wheat. The enzymes from the yeast are going to break down the starch from the dough. Initially, the reaction is going to be aerobic, but as oxygen is used up, it could occur anaerobically. CO2 is going to be produced, and this is going to aid in that expanding or causing the bread to rise. So I said yeast contains amylase enzymes that break down starch. Yeast can respire both aerobically and anaerobically. Initially, as yeast is mixed in the dough, aerobic respiration takes place, producing carbon dioxide and water. After the oxygen runs out, anaerobic respiration takes place, producing carbon dioxide and ethanol. And as the baking begins, ethanol evaporates and the trapped carbon dioxide escapes, causing the dough to rise. So this brings us to the end of question 7. Let's continue to question 8. In question 8, the diagram shows the female reproductive system and the male reproductive system in some structures labeled. So they say which structure produces gametes. Gametes are going to be produced from this structure as well as this structure. So since the ovary is not labeled for females, I'm going to go for males with the testes. And then next they say which structure is the site for fertilization. fertilization occurs in the fallopian tube or the oviduct, and that is part A here. And next they say, in which structure does the placenta usually develop? The placenta develops in the uterus, which is structure X. So X is the uterine wall. In part B, some males have an operation called a vasectomy. This means that they do not release sperm. This involves the cutting and sealing of sperm ducts, which is the vast difference inside the man's scrotum. On the diagram, draw two lines to show where the cuts are made. The cuts are going to be made along this point here because the sperms are made from in here and they will go upwards. So if the cut is made here and here or along the tube, sperm cannot come up in order to be released in the outside of the male's body. Next they say, explain why this operation can be used as a permanent way of preventing pregnancy. This is because cutting prevents sperm release, so the semen will contain no sperm during ejaculation, so no sperm will enter through the cervix of the female, and fertilization of the egg will not occur. In part C, female fertilization is another operation that can be used as a permanent way of preventing pregnancy. This involves cutting and sealing the fallopian tubes, which are the oviducts. Scientists compared female and male sterilization looking at the outcomes of the operations. This is their data. So when we look at the number of deaths during the operation, in female sterilization, this is per 100,000. We see 2.29, which is greater, and males we see 0. The number of deaths following the operation, the males are 0, is high in females. The number of major complications are higher in females in comparison to male. The number of sterilization failures are higher in females, and the short-term costs in millions of US dollars, it's more expensive in females in comparison to males. The scientists concluded that male sterilization should be recommended for most couples considering a permanent way of preventing pregnancy. Discuss this conclusion using data from the table. Of course, we're just going to discuss this. It's quite easy. Male sterilization has no deaths during and following the operation, but female sterilization has. The number of major complications is higher in females than in males. The number of sterilization failures is greater in females. The cost of the operation is higher in females than in males. And since data was collected for 100,000 people, this is a large study and the results are reliable. So this brings us to the end of question 8. Let's continue to question 9. In question 9, farmers and scientists have worked together for the last 50 years to develop crop plants and farm animals with desired characteristics. Explain why selective breeding with crop plants is easier and quicker than selective breeding with farm animals. If you compare animals and plants, usually animals have fewer offsprings, and it takes longer in order for the characteristics to be observed in the phenotype of the offsprings. However, plants mature within a shorter period of time and they have multiple offspring, so it's easier to study using plants. So I said plants mature faster, so the results from one genetic cross are observed faster, and plants produce numerous seeds or offsprings, so a lot is available to select from for the next cross, and the conditions for plant growth can be easily monitored and controlled than conditions for animals. In part B, selective breeding in cattle has been used to improve features such as higher milk yield, longer life, and increased muscle mass. Describe how selective breeding can be used to improve milk yield in dairy cattle. For any characteristic that you want to improve using selective breeding, you have to select organisms that have the best or related qualities or organisms whose parents had those qualities. So here I chose to select a female whose mother produced a lot of milk and select a bull whose mother produced a lot of milk. Then mate the selected cow and bull to get the offsprings, and then from the offsprings, breed a cow that has the greatest milk production with a selected male, and then repeat this for many generations. In part C, scientists and farmers are also selecting animals based on behavior and temperament. Suggest why an animal with a calmer temperament may be more suitable for the farmer on a dairy farm. Animals with a calmer temperament are going to be easier to handle because they will not be aggressive and it will be easier for them to be milked. So I said those with calmer temperaments will be less aggressive and have less chance of harming other cows or harming themselves or even the caretakers. They will be easier to manage and it will be easier for them to be milked. This energy will be wasted in fights, so more milk production will occur, meaning energy is going to be used for milk production instead of being wasted on their aggressive behavior. Next, I say, cattle behavior is also affected by the hormone adrenaline. When cattle are more stressed, they have higher levels of adrenaline. The effect of adrenaline on the cattle nervous system is similar to the effect of adrenaline in humans. Describe the production of adrenaline and its effects on the body. Adrenaline is responsible for the fight or flight response, meaning preparing the body for activity. So I said adrenaline is produced from the adrenal glands to prepare the body for activity, which is the fight or flight response. Adrenaline causes the heart rate to be increased. Adrenaline stimulates the conversion of glycogen to glucose, and it causes the diversion of blood away from the gut towards the muscles. Adrenaline increases the ventilation rate. It causes dilation of the pupils, and in some animals, it causes the body hairs to stand, making them appear bigger to the enemy. Stress in cattle can also affect their reproductive system, leading to damage to the placenta. Describe the role of the placenta in reproduction. The placenta enables movement of substances between the mother and the fetus. These substances are going to be either nutrients as well as oxygen from the mother to the fetus, or a movement of waste from the fetus to the mother to be taken out of the body. So I say the placenta allows for the diffusion of substances to and from the fetus. It delivers oxygen, nutrients, and antibodies to the fetus and removes carbon dioxide and urea or waste from the fetus. The placenta also secretes progesterone hormone, which maintains pregnancy. So this brings us to the end of question 9. Let's continue to question 10. Question 10. Plants absorb light energy for photosynthesis. Some colors of light result in a higher rate of photosynthesis than other colors. Devise an investigation to discover which color of light results in a high rate of photosynthesis. Include experimental details in your answer and write in full sentences. So here we are varying the wavelength of light because they're talking about color of light. Different wavelengths have different colors. In my description, I set up an experiment using a water plant. The plant is going to be exposed to different colors of light, meaning using different bulbs of light. And then all the other variables like temperature, carbon dioxide concentration, and so on are going to be maintained as we vary the wavelengths of light or the colors of light. And then I'll repeat the experiment for each color in order to calculate the mean number of bubbles of gas produced. So I say set up the experiment using light bulbs of at least 5 different colors. Use a water plant like a pondweed. And for all experiments, the plant should be of the same species and age. And if possible, use the same plant. And here, if you decide to use the same plant, you must give the plant time to acclimatize or get used to the new conditions before you begin to count the bubbles of gas produced. Otherwise, your results are going to be faulty. Repeat the experiment for each variation in light color and calculate the mean. For the dependent variable, we count the number of bubbles of gas produced per time. This could be 10 minutes to determine the rate. And then control variables like temperature, light intensity, carbon dioxide concentration, pH, mineral ions, and so on have to be controlled in all experiments to be the same. The concentration of carbon dioxide has to be provided using sodium hydrogen carbonate. Since I used a water plant, this should be constant and in excess so that the concentration of carbon dioxide does not limit the rate. In this experiment, the light color that gives more bubbles is the one that gives the highest rate of photosynthesis. So this brings us to the end of question 10, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.